today my topic for presentation is morphological characteristics relevant in understanding fungal pathogens. So as we are not used to the current names which have been changed, so I have used common names which we use in routine for this presentation. So I will start my presentation. First, we should know some definitions, basic concepts about the fungi. What is fungus? Fungus is a eukaryotic unicellular to filamentous chlorophyllous organism having an absorptive nutrition which multiplies sexually and asexually or by both methods. It can be either yeast or mold. So the yeast, what are yeasts? These are actually unicellular fungi consisting of spherical, round or elongated cells that propagate by a sexual process called as budding. Here you can see these are the budding cells. Either it can be broad-based or it can be a narrow-based budding, or it can produce a pseudo hyphae, or it can be having these cells can be having septum. So this was about yeast. Then another form is mold. Mold is formed of the mycelium. So here you can see this is the mycelium. Mycelium is mass of intertwined hyphae. So first, basically, you should know what is hyphae. Hyphae, these are the hyphae. These are tubular branching filaments constituting basic fungal growth unit that makes vegetative or aerial mycelium and increases in length as a result of apical growth. So these hyphae can be divided by a septa. Here, these are the cross walls. So they are divided by the septa. They are depending upon the presence or absence of septi, it is called as septate hyphae or aseptate hyphae. Hypha again can be termed as a hyaline hyphae or fluid hyphae. Hyaline hyphae means in the microscopy they will be looking clear, transparent and colorless. That is hyaline hypha. If they are having dark pigmentation like brown color or black color pigmentation, then they are called as fluid hypha. Then you should know what is vegetative mycelium and aerial mycelium. Vegetative mycelium means the fungal hyphae which are embedded in the culture medium are called as vegetative mycelium. So these are responsible for the absorption of nutrition. And another are aerial mycelium. These are above the culture medium, and they consist of conidia. So, and you should know what are pseudohyphae. Pseudohyphae are produced by the budding cell, but that is an elongation of a parent cell before it buds, and as a result, it results in the chain of elongated cells. So that's why it is called a pseudohyphae. So all the fungi will produce spores. So what is spores? The fungi will reproduce by means of macroscopic propagules, which consist of a single cell or several cells contained within rigid cell wall. So the spores can be produced by either asexual reproduction or sexual reproduction. Asexual stage is called as anamorphic stage, and sexual stage is called as telomorphic stage. So sexual reproduction can be of two types: homothallic or heterothallic. Homothallic sexual reproduction means the homothallic means it is a self-fertile, where asexual reproduction, where sexual reproduction can take place within one thallus, and in heterothallic they are self-sterile and requiring two compatible thalli for sexual reproduction. In general, sexual reproduction is difficult to obtain in the culture. Hence, most of the fungi are identified by asexual reproduction characteristic features. So, sexual structures can be formed in the Asomycota, Basidiomycota, Mucormycotina, and Entomopteromycotina. So in Asomycota, asocarps will be formed as sexual structures. So asocarps, these are asocarps. These are fruiting bodies which bears asci. What are asci? Asci are round or elongated sac-like structures which usually contains 2 to 8 asospores. And asospores are the sexual spores produced in a sac-like structure called as ascus, called as asospores. So asocarps is divided into two types: perithecium and clistothecium. Perithecium means it is a large, round or pear-shaped structure, usually having a small rounded opening called as ostiole and containing asci with azospores inside. So the perithecium will release its spores through an apical opening called ostiole. The example which produce the perithecium is Tutuminium species, and the type is Clistothecium. Clistothecium are a large, fairly round, closed and multicellular fruiting body in which randomly disposed asci and spores are formed and held until the structures burst out. It will split open to liberate its content. The examples for the fungi which produce Clistothecium are Aspergillus nodulans, Aspergillus glaucus. Next, in the Basidiomycota, Basidiocarps will be produced as sexual reproduction products. So Basidiocarps are the fruiting bodies where basidia are arranged inside the Basidiocarp. They are macroscopic structures. What is basidium? Basidium is a single club-shaped cell which bears the basidiospores. It's called a basidium. Basidiospores are the sexual structures found on the basidium in the Basidiomycota. The example for this is the sexual stage of Cryptococcus species. Next, we should know about the Zygospores. Zygospores are produced in the Mucormycotina and Entomycormycotina. So here you can see the zygospore. These are the fusion of multinucleate tips of two hyphae leading to formation of a single large zygospore lying between them. This is a multicellular multinucleate thick-walled structure that has evolved to endure the adverse environmental conditions. So here you can see this is an intercalary zygospore seen in the Basidiovelus species. So to understand the morphology of the fungi, we should know the process of conidiogenesis and the sporogenesis. So what is conidiogenesis? It is the process of asexual spore production. It is called as conidiogenesis. What is conidiogenous cell? The fertile part of the cell that produces conidia is called as conidiogenous cell. What is conidium? Conidium is a asexual unit formed by budding of tips of hyphae or from the walls of the hyphae. And the conidiophore. These are the specialized structures bearing conidiogenous cell. These are called as conidiophores. So conidiogenesis can be of two types, thallic method or blastic method. So here you can see this is a blastic method, this is thallic method. Blastic method means in this there will be a repeated budding that permits them to produce large number of asexual spores or from a single conidiogenous cell. So here there will be a budding, repeated budding to produce large number of asexual spores called conidia. But in thallic method, the existing hyphal cell is converted into the conidia. This is the difference. So conidiogenesis broadly it is classified into these types. So in the blastic, it can be enteroblastic or holoblastic. In enteroblastic conidiogenesis, again phyloidic method and analytic method. In holoblastic, there will be a synchronous holoblastic conidiogenesis and sympodial holoblastic conidiogenesis. In thallic method, it can be holothallic or thalliarthric. In thalliarthric, it can be again divided into three types: holoarthric, enteroarthric, and endogenous arthric. So first, you should know about the enteroblastic and holoblastic. What is the difference? Holoblastic means it involves all the cell wall layers of the conidiogenous cell for conidia formation. All cell wall layers are involved, but in enteroblastic, the conidia are formed from the inner cell wall layers of the conidiogenesis. That's why they are called as enteroblastic. You should know certain terms for the understanding the conidiogenesis process, like what is catenate, what is acropetal, and what is precipitate. Catenate means these are the end-to-end arrangements seen in chains of 3 or more spores. Here, this is a catenate arrangement. And if they are dissolution, they are split after formation, then they are called as non-catenate. So acropetal arrangement. Acropetal arrangement means this is acropetal arrangement. The conidia developing from the base towards the apex, wherein the youngest is at the top of the chain. For examples are Alternaria, Cladosporium, and Curvularia species. Then basipetal arrangement. the youngest conidium is at the base of the chain. So it is called as basipetal arrangement. It can be seen in the Aspergillus and Scopulariopsis form. So we will see first enteroblastic conidiogenesis. It is again divided into two types: phylotic enteroblastic conidiogenesis and analytic enteroblastic conidiogenesis. Examples of phylotic method are Phyalophora species, Aspergillus species, Penicillium species. Examples of analytic method are Scopulariopsis species, Exophyala species. So what are phyllites? So there are phyllites are the type of conidiogenous cells, usually flask shaped, which produce basipetal succession of enteroblastic conidia from apical opening without resulting in change of the length. There will not be any change of the length, but they will produce the conidia. It can be of two types: slimy non-catenate or dry catenate stage. So slimy non-catenate means the conidia will be released after formation of single conidia. In this collarate zone will be formed because inner cell wall layer, outer cell wall layer will be there. That's why collarate zone will be formed. This is slimy non-catenate, and dry catenate means each initial conidium forms a new wall chain. So these are dry catenate stage. So the example for dry catenate conidia with phyloidic formation are Aspergillus. We should know the parts of Aspergillus first. So here these will be forming separate hyphae. The conidiophore will be arranged on the which arised from foot cell. They are also called as types. So this is a conidiophore which will end into the swollen vesicle. The vesicle will be embedded in the vesicle will be a swollen end where the phyllites and metallo will be there, will be formed. So if in Aspergillus there are two terms, uniserate or biserate. Uniserate means only phyllites will be present. In Aspergillus, conidia will be formed by phyllide only because it is phylladic. Phyllites are the conidiogenous cell in Aspergillus. Hence, if it is uniserate, only phyllites are present, and if it is biserate, first on the vesicle medulla will be present, and then above that phyllites will be present, and from phyllide the conidia will be formed. Then another two important terms in Aspergillus are columnar arrangement and radiate arrangement of conidia. So if this is a columnar arrangement, actually in this compact closely placed phyllates that are parallel to the axis of conidiophores will be there. So that will be a compact arrangement. It is called as columnar, and radiate means spreading out in all directions from the center. It's called as a radiate arrangement. So in Penicillium also phyllates medullae are present. So there will be staging. Either it can be triverticillate, biverticillate, or monoverticillate. So depending upon the species of the Penicillium, it will be forming triverticillate or biverticillate form. Another method, another examples for phyloidic conidiogenesis are the Phyalophora and Acromonium, but they are non-catenate. Here you can see they are non-catenate. These are the phyllites. These are the phyllites, and these are the conidia. Now we will see what is analytic enteroblastic conidiogenesis. So what are annelids? Annelids means the conidiogenous cells where the conidium is formed from all layers of first way of conidium will be formed from the all layers of an elongated apex of conidiophore and subsequently from the inner cell wall layers in a basipetal manner. So it will form the annellated zone. Morphologically, it is distinct because of apical rings composed of the outer cell wall remains at each released conidium. That is, annellated zones will be there. So annelidic form can be seen in the Scopulariopsis brevicularis. Here these are alienated rings will be there, and here also in the Exophyala we can see the annulation. So these are the annelids which will produce the conidia. So these are Enteroblastic conidia in the dry chains and in wet masses. In dry chains, the examples are here in the aspergillus. We can see the phyllites and these are the annelids. In wet masses, these are the phyllites and these are annelids. So now we will see holoblastic conidation. It is again divided into two types: synchronous holoblastic conidiogenesis and sympodial holoblastic conidiogenesis. So synchronous holoblastic means at the same time all the budding will be formed for producing the conidia. That's why it is called as synchronous. But in sympodial holoblastic conidiogenesis, first one conidia will be formed fully, then it grows past to it, and again second budding will be formed. Then after formation of second, third budding will be formed. So it is a sequential process. So it is called a sympodial holoblastic conidiogenesis. It can be seen in the curvularia species, bipolar species, and alternaria species. So holoblastic conidation can be present like a single holoblastic conidium, or it can be presenting in the chains, or sympodial means on the geniculate conidiophore. So how it will produce the chain? The example is Cladosporium species. The actually formed conidium produces a second conidium, and then second produces a third, and so on, until the chains of the spore is produced with the youngest is at the tips. As each conidium can produce more than one bud, a branching chain becomes possible. So example is Cladosporium species, and genetica some fungi will produce geniculate conidiophore. This is the geniculate conidiophore. The conidiogenous cell that produces the first formed spore then grows past to it, produce second one. If this process is repeated, and it will result in an elongated conidiogenous cell called as geniculate conidiophore with numerous lateral single spores to its sides. The examples are Alternaria bipolaris. So here these are some examples. Here Alternaria, you can see sympodial growth. Here also geniculate cells are there, and this is curvularia. So this is very important, sympodial holoblastic formation. In this conidiogenous structure that continues to increase in the length by forming a new growing point just below each new terminal conidium, often resulting in the geniculate appearance. So this was about the blastic. Now we will see the thallic conidiogenesis. Thallic conidiogenesis is again divided into holothallic and thallic arthric conidiogenesis. Thallic arthric is again divided into holoarthric, enteroarthric, and endogenous arthric conidiogenesis. So holoarthric conidium development. In this, we should know the term rhizolytic. Rhizolytic means the separation of the cells either by the lysis or fracture of the cells. It's called as rhizolytic. So holothallic means in this from this conidiogenous cell, the septations will be developed, and after maturation, the cell will dissolve by the lysis or fracture. So it is holothallic conidium with rhizolytic conidium liberation. So the example, the septations will be formed. The examples are the macrospores formed in the dermatophytes. These are the some examples, and these are fractured from the original hypha. So next is holoarthric development. Holoarthric development means this is the conidiogenous cell. Here the arthrospores will be formed, and schizolytic term. The schizolytic means they will be released by production of the double septum. Double feature will be there. So there will not be any lysis. The formation of double feature, and after that it will be getting detached. So this type will be occurring in the Geotrichum species. Next is enteroarthric conidation with rhizolytic conidium liberation. So this can be seen in the coccidiot species. So in this, this is a thallus conidiogenous cell. In this, inside the arthrospores will be formed, but the release of the arthrospores will be done by the alternative lysis of the cells, some parts of the arthrospores. So This is enteroarthric. Then endogenous. Endogenous conidium development means this is a conidogenous cell. Inside that, arthrospores will be developed fully, and then at the end, the conidogenous cell will be ruptured to release the all arthrospores. The example is Basidiocrp. So, by thallic method, arthrospores can be produced and chromatospores can be produced. So there is one term called as aliridospores. Aliridospores are the intermediate state between the thallic and plastic conidiogenesis. The thick wall terminal or lateral single cell conidium attached by wide base and released by the lysis or fracture of supporting cell. They are bound directly on the hyphae and short pedicles or undifferentiated conidiophores. These can be seen in the dermatophytes. And the aliridophores are formed on the conidium, aliroconidium, but commonly conidium tub is used. So we should know the structures in structures which are present in the mucormycetes. So first we will see from the hyphae there will be a sporangiophores. Sporangiophores will be formed. Sporangiophores will end into the columella, which will be entering into the sporangium. This is a columella. Just below columella, there will be a apophysis, and sporangiospores will be formed in the sporangium. So, and there will be a rhizoids just below or in between the two stolons. So, and there are merosporangia, which are produced in the Syncephalastrum species. Merosporangia means the sporangiospores are arranged in a row. They are called as merosporangia. Here you can see these are merosporangia. Here this is a sporangium, and this is columella. So sexual stage of Mucormycotina includes Zygospores. Next, rhizoids. Rhizoids are actually root-like branched hyphae extending into the medium. They can be produced by various species like Rhizopus, Rhizomucor, Listhemia corymbifera, Apophysomyces elegans, and Saxenia vasiformis. So next, we should know the Chlamydoconidium. So these are enlarged, rounded conidium that is thick-walled and contains stored food. They can be served as a survival propagules. So terminal Chlamydoconidium, then intercalary Chlamydoconidium, and the Chlamydoconidia in the chains. These are the three types. So in terminal, the Chlamydoconidium will be at the end. It can be commonly seen in the Candida albicans. Then intercalary Chlamydoconidium can be seen in some dermatophytes like T. violatium, T. tonsurans, M. ferruginum, and in vitro hertapsa we can see. And Chlamydoconidia in chains can be seen in T. verrucosum. Next, we should know about the Arthroconidium. Arthroconidium, these are an asexual spores formed by breaking off of a hyphae at the point of septation. They are rectangular or barrel shaped. Examples are Trichosporan species, Geotrichum species, Malbranchia, Neocycalidium, and Oxidioid species. So we should know about the structures Macroconidia, Microconidia. Macroconidia are usually multicellular large conidia. There are some fungi which produce only Macroconidia, like Alternaria, Bipolaris, Turbularia, Eulocladium, Epidermophyton, Flocosum. And some fungi will produce both macro and microconidia. Macroconidia means they will be mostly single celled. They can be oval or round or pear-shaped structures. So here you can see the Macroconidia fusarium species. These are in the dermatophytes. These are the Macroconidia. And these are the Macroconidia of the histoplasma species where tuberculate knob-like structures are seen. So some dermatophytes will produce spiral hyphae, which are corkscrew-like turns of mycelium. They can be present in the T. mentophytes and tonsills. So some dermatophytes some fungus will produce fabric chandelier. The fabric chandeliers are the many short, multiple branches at the end of the hyphae, resembling reindeer horn or chandelier. So the examples are T. shanleni, T. violation, and hot burning. Some fungi will produce Racquet mycelium. Racquet myceliums are the localized swellings formed by tightly twisted hyphae resembling tennis rackets. The examples are Microsporum species or Efloccusum species can produce Racquet mycelium. Lastly, Hulle cells. These are the thickened, large sterile cells with small lumens. They are associated with Piscothecia produced by sexual stage of some fungi. The examples are Aspergillus nidulans, Aspergillus glaucus, Aspergillus pallidostus. These can form the Hulle cells. So today, in a given time, I could describe only these morphological features. If there are any queries, you can